Tonight it's my pleasure to introduce to you Deneen Wellik. She's the new chair of the Department of Cell and Regenerative Biology here at UW-Madison. She was born in Washington, Missouri, and went to St. Francis Borgia High School there in Washington, Missouri. Then she went to Washington University and got her undergraduate degree in biology. Then she came to a place called Madison, Wisconsin to work at Hazelton Labs out by the airport. And then she went to grad school here to get her PhD in biochemistry under Hector DeLuca. She postdoced with Mario Capecchi at the University of Utah, and some of you may recognize him as one of the 3 people that won the Nobel Prize in Physiology or Medicine in 2007. We're pretty pleased that Oliver Smithies was one of the other 3 people, and the work for which Oliver Smithies was awarded was done in the building next door to us, there at 445 Henry Mall, otherwise known as Old Genetics. And then she went to the University of Michigan and was there for how many years? 15. And then in December of last year, 2018, she came back to UW-Madison to take her current position as chair and professor in the Department of Cell and Regenerative Biology. As I get older, it's always interesting to ask, what's it take to make a new muscle cell? I'm not sure she's going to address that, but I hope she does, at least a tiny bit. Because I can't figure out how to do it myself, so maybe you can. Would you please join me in welcoming Deneen Wellik to Wednesday Night Live?
Hi, thanks for the opportunity to talk to you tonight, and I actually won't specifically address the muscle. I'll address at the end. Part of what I took out of the talk to not have too much in tonight is how these same genes and cells do operate in the muscle. What I'll talk about tonight is the skeleton, and in general the cells that we'll touch on that I would not have expected to do so early in my career are what I'll try to convince you are in fact musculoskeletal stem cells, but are also referred to as MSCs. And how I got to that point, which was not by trying to study those cells. What set of genes that I've been interested in ever since beginning my postdoc is a set of genes called the Hox genes. I have always studied them in mouse, but they were discovered in the fruit fly Drosophila in the late 1970s is when they're first reported. And they had several really interesting characteristics, particularly to developmental biologists, which is what I do. And through a series of creating random deletions along, this is the polytene chromosome of a Drosophila, what mostly Ed Lewis, but also other investigators found is that when mutations happened, I'm not necessarily pointing to the right region, but deletions happened right in a row, indicating to them that these genes were all in a line. What they found is that both the expression and the function of these genes along the line of the chromosome was reflected in how they patterned the developing Drosophila embryo. So they were expressed from head to tail in order along the chromosome, and loss of function of each of these genes caused really dramatic patterning defects that were termed homeotic transformations. And I'll show you a couple of the more dramatic examples of the patterning here. With loss of function of this Hox gene UBX, what you can see is an example of what happened in any of these cases. In this case, hopefully you can see the normal third thoracic segment of the Drosophila has these little teeny balancing wings called halteres. And this is right behind the second thoracic segment, which has the full complement of wings, or 2 wings, 1 on each side. So when the function of UBX was lost, what you can see is that this third thoracic segment didn't develop into these halteres, but rather made a duplication of the segment that was just anterior to it. And in fact, the loss of function of any of these genes caused an analogous transformation of the segments along the Drosophila, showing that these genes, at least in the fruit fly Drosophila, were really critically important for patterning along the AP axis, the anterior-posterior axis of this animal. Likewise, when these genes were overexpressed in regions they weren't normally expressed, the example shown here is this gene, antennapedia, now being expressed in the head region that's normally expressed lower in the body. What you can see now is the Drosophila, which normally has these antennae, that are on its head are now growing pretty perfectly shaped legs in the place where their antenna should be. So again, really supporting that these genes were critical to patterning the body plan of this animal. So in the 1980s, as genes started to be cloned out of both mouse and human, it was found, not surprisingly, that these genes were conserved through mouse and through human. And this is actually a picture of all 39 of the Hox genes that are in all mammals. The exact same complement of these 39 genes exist in mouse and in human. And they're analogous to the genes shown in Drosophila, as is reflected here. And included in the conservation is not just the genes themselves, but actually their collinear arrangement along the chromosome. So throughout evolution, these genes have stayed conserved both in their general sequence, but also in their arrangement and their expression along the anterior-posterior axis of the body. So I was working on retinoids in Hector's lab here as a biochemist when the retinoids, or retinoic acid, was being found, just reported in the 90s, to cause changes in expression in the set of genes. So that's actually how I started reading about them. And I thought, this is so fascinating because we didn't really know what these genes did. And it was happening right at the time when Mario and Oliver and other people were learning the technology to make genetic mutations. And what Mario's lab decided to do, or what Mario decided to do with this newfound technology, is knock out every single one of these 39 Hox genes, which his lab did really in the course of 5 or 6 years at the University of Utah, just prior to my coming. So when I started in his lab in 1996, they were celebrating the germline mutation of the 39th of the 39 Hox genes. Really an unbelievable accomplishment from the 1990s. So as someone interested in these genes, that was really exciting. To Mario, I think this was a little less exciting because he picked these genes thinking they would be the panacea of understanding how the vertebrate body plan was laid out. And in fact, when each of these genes was knocked out individually, most of the time, very little happened. In fact, in some cases, they couldn't detect a phenotype at all. So in some ways, this was relatively disappointing to him, but the toolbox was there for someone who wanted to look at these more carefully. And so the key to this particular mystery wasn't that the function of these genes weren't conserved. It's that the conservation through evolution actually protected them so much from being mutated that these paralogous genes, as they're called, so the genes that are most closely related to each other in the same place along the chromosome, actually retained a lot of what's called functional redundancy. So if you knock out this gene alone, HOXA10, really nothing happens. If you knock out HOXA10 with HOXC10, and that's 4 mutant alleles, really nothing happens in the animal either. But if you knock out all 6 copies of these 3 related genes in the HOX10 paralogous group, and this is again just an example that happened throughout the axis, you can see now, hopefully, this very dramatic patterning phenotype. So this is a picture of the ventral side of a newborn mouse skeleton. You can see the end of the thoracic or ribbed vertebra, the lumbar vertebra, sacral, and then the beginning of the tail. When you lose all 6 copies of these HOX10 genes, what you can see is that the ribs don't stop at this 13th thoracic vertebra, but instead grow throughout the rest of the lumbar and even the sacral skeleton. And I should say, if any 5 of these 6 alleles were knocked out, what we saw in that case was one little teeny blip of an extra rib. So that's how much redundancy is retained within this group. So we spent the next several years knocking out all of these paralog groups and showing that indeed the patterning along the skeleton was conserved and controlled by this set of Hox genes. This was work done by Moises Majo's lab just a couple of years after we published this phenotype. And after seeing these results, he took one of these 10 genes, it was HOXA10 in fact, and created again this gain-of-function mutation. So instead of being expressed at this level of the embryo and posterior, he expressed it all over the whole entire embryo. And you can see in this case, the ribs, and in fact you can't see here, but also the sacral extensions are completely gone. So again, this really reinforced the idea that these Hox genes really have conserved patterning roles in mammals as well. And this has been a question in my lab in terms of how does this happen we've been trying to address. So in vertebrates, in limbed vertebrates, this posterior part of the HOX gene, the HOX9 through HOX13 genes, have been co-opted, so they're also expressed along the proximal distal axis of both sets of limbs, the forelimbs and the hindlimbs. And this is an example of the phenotypes you see, again, with the paralogous loss of function mutants. In the HOX10 genes, hopefully you can see that this, in this case, the femur, and developmental biologists will refer to this as a stylopod because that means you can refer to the forelimb and hindlimb identically, is dramatically mispatterned, but the zeugopod, or the tibia and fibula in this case, are fine, as is the autopod, or the foot. With loss of HOX11 genes, you can see that now the zeugopod region, here showing the radius and ulna, is dramatically mispatterned. And with loss of the HOX13 genes, you can see that the animal, it has a very mispatterned autopod. So this developmental patterning transcription factors have been used also in the proximal distal patterning of the limb. And we have used this particular system, the zeugopod, and the HOX-A11, D11 genes specifically that lead to this phenotype, to try to explore how this happens in mice. And the reason is not that we're particularly interested in the radius and ulna as opposed to the rest of the bones in the body, but by a fluke of evolution, one of the three HOX genes, HOX-C11, has lost its expression in the forelimb. So this phenotype is created with loss of only 2 genes, or 4 alleles. So genetically, this makes it a lot more tractable to pursue than having to knock out 3 or 4 copies of the gene to look at. So we have focused on this with the hopes that what we find about these genes would be relevant to the rest of the HOX genes as well. And our question really has been interesting, we've been interested in is how do HOX genes pattern along the entire skeletal axis of mammalian embryos. So the first new allele, or new gene-targeted mouse that we made in my lab when I started at the University of Michigan was a GFP insertion allele into the HOX-A11 gene. And actually, I want to point out here, and for the rest of the talk, if anyone has questions during the talk, please feel free to ask them. Don't wait till the end. If it helps you, understand what I'm saying, I'm happy to take questions anytime. But so this allele, this HOX-A11 GFP allele, has a green fluorescent protein, that's what GFP stands for, that's inserted in place of the HOX-A11 gene. So wherever HOX-A11 is normally expressed, instead you will see this, be able to detect the expression of GFP. And we looked at this then during limb development and found that right after its expression starts, where it was already known by in situ that all of these posterior HOX genes are expressed together right as the limb bud forms, but as soon as the elements of the skeleton start condensing, you can see that these HOX11 genes become confined just to this zeugopod or radius and ulnar region. And if we section, just take sections through that developing skeleton, hopefully what you can see here was the first very surprising result from this new allele. The green staining you see here is the GFP expression. The red staining is actually SOX9, which is one of the earliest factors that's expressed in condensing cartilage. So the cartilage being the material that preconfigures the skeleton. What hopefully you can appreciate here is as soon as you start seeing condensation of these 2 zeugopod elements, the HOX genes are not expressed there at all. They're expressed instead immediately surrounding these cartilage elements. And this was surprising, of course, given the dramatic phenotype in the cartilage and bone elements. This was surprising. And in looking at this in a little more detail, not only is the HOX expression not in the cartilage, it also isn't expressed in the developing osteoblasts. The osteoblasts being bone-forming cells, the cells that replace the cartilage with bone, also don't express HOX. If you look very closely, the HOX expression is just outside these osteoblasts. So that was very confusing and frankly a little disappointing because now we're left with a set of transcription factors which can only regulate gene expression as far as we know inside the cell, not being expressed in any of the cells in which it has a phenotype. So this was a mystery and remained a mystery for a pretty long time. The second surprising thing, and in retrospect I can't tell you exactly why it was surprising to us, is that when we section through these embryos all the way out to newborn stages, this is just the day before a mouse is born, this expression is still here. So developmental biologists had really thought of these genes as early patterning transcription factors, and I think we didn't imagine that they would still be expressed at this late of stage, and that was curious to us. And this finding is what led us in a new direction that I think has led us to understand these genes a bit better. Oh, great. So this is a project that was taken up by Danielle Rux, who is a former graduate student in my lab. She defended a few years ago. And she took this GFP-positive animal and she just started looking later after birth and found that indeed these genes stay expressed all the way through the life of the animal. They maintain some similarity to embryonic expression. This is just using an antibody. That's why it's blue to the GFP. This is GFP expression. You can see it's still in the outside of the bone, what's called the periosteal region, after the bone starts developing. But also, perhaps you can see that it starts being expressed on the inside of the cortical bone, or the endosteal region. And if you look closely at the bone marrow region, you can start to see also, and hopefully you can see from there, expression within the bone marrow. So that was really interesting, and this is a picture of that a little closer up, a very close-up picture of the bone marrow. Hopefully you can see, in addition to the outside of the bone, the inside of the bone, also you can see this spicularly looking expression in the bone marrow of the bone. And a collaborator and a colleague of mine at the University of Michigan who worked on hematopoietic stem cells said, those look like MSCs. They're really in the exact right proportion. They have the right shape. You should really check and see if your genes are being expressed in these adult MSCs or mesenchymal stem cells that people had been looking at. And so mesenchymal stem cells, or MSCs, is perhaps one of the worst terms that's ever been used in biology, actually. So MSCs are used to describe by some clinical and engineering folks who use them for tissue engineering applications as anything you take out of the bone marrow, throw onto a tissue culture plate, and that it grows. So in fact, MSCs that are being used for clinical therapy and sometimes in bioengineering approaches are really just those cells that grow out. They stay growing in culture, and they've used them for lots of interesting things, but they're not very well defined. People that work in both human biology and mouse biology, and this is not work by my lab, have really spent the last 10 years or so trying to identify that proportion of the stromal cells that are in the bone marrow that actually have progenitor activity. And that's been looked at by several ways and has been used to refine what people refer to as progenitor-enriched MSCs in the bone marrow. Some of the ways they look at this are cell surface markers that are on the surface of these cells. PDGFR alpha and CD51 are two that are used in combination that really enrich for progenitor activity among the stromal cells that grow out of the bone marrow. LEPR is one. There have been mice made that are genetic lineage traces that specifically label populations that are enriched for these progenitors. Another thing progenitor-enriched MSC should do in culture is show colony-forming unit activity. So if you take these cells out and grow them, you should be able to take anything that's a progenitor individually and grow it up into another colony and have it keep growing in culture and keep dividing and self-renewing. And those cells should also be able to differentiate. Any one of those cells grown up into a colony should be able to differentiate into cartilage, into bone, and into fat. That's one of the main characteristics that shows that at least in vitro they have progenitor activity. And also, they should expand when you fracture the animal. So when the bone is fractured, it's those progenitor-enriched cells that expand to heal your skeleton. So there were lots of ways we could ask, are our HOX-expressing cells in this population? And so that is what Danielle did, and the results were very exciting. So what hopefully, I can't see very well here, but hopefully you can, is that if you take all of the bone marrow cells from the zygopod of a mouse, 98–99% of them are some kind of blood cells. And HOX genes are not expressed in the blood cells, they're expressed in the non-hematopoietic portion, or the 1 or 2% that's not your blood. If you take this 1 or 2% and look at it further by cell surface markers, what you can see is that some of these stromal cells, now this from the small percent, are endothelial cells or vessel cells in your bone marrow. And HOX is not expressed in the endothelial cells. They're rather in this non-endothelial component from this 1 or 2% of the bone marrow. Now, if you take this non-endothelial population and sort further, you can take this population and sort it on PDGFR alpha CD51, for instance, and it is only the 6 or 7% of these cells that express both of these markers that other researchers have shown are then enriched for this progenitor activity. And hopefully what you can see is not only are some of our GFP cells in this double positive quadrant, really virtually all of them are only in this tiny fraction of cells. This is showing the two markers individually just to show how completely they segregate with these double positive markers. And then this is another marker, LEPR, along with a genetic lineage trace of the expression of that known MSC marker. And what hopefully you can see here is that this is the LEPR Cree rosa tomato that had been previously published to be expressed exclusively in this progenitor-rich MSC. This is the GFP expression, and if both of these things are expressed in the same cell, you'll see these cells turn yellow. So what I hope you can see here is that all of the green cells are also red or turning yellow, but not all of the red cells are green. And that's an important distinction. And again, this is just the GFP cells, again, sorting far into that LEPR positive fraction. And I'll come back to this point, actually, I think in this next slide. So if we take the GFP positive cells from the bone marrow and put them into culture, what we can show is they do trilineage differentiate clonally. So they can make bone, they can make cartilage, and they can make fat. What this assay shows is the CFUF assay. And again, other people have shown if you segregate this tiny population for this PDGFR alpha CD51 double markers, you can show that they have clonal regenerative capacity in vitro. And this is about how much. So if we take the half of this population that also expresses GFP and do the same assay, you can see they have 3 times as much clonal activity in vitro. It's an in vitro assay, but it was really pretty exciting to us because it suggested that we might really have a cell population that has the progenitor activity. So this was awesome. These cells also expand in response to fracture. Hopefully you remember the expression was just along the lining of the bones. When you fracture the bone, you see this giant expansion. These expanded cells stay lineage labeled in all the ways one would expect for a progenitor cell. And so in this paper that we published a few years ago, what we reported was that our HOX-expressing cells are, in fact, progenitor-enriched MSCs. They have the right cell surface markers, they genetically lineage trace with the right things, they have CFUF activity and trilineage differentiate, expand in response to fracture, but they have 2 other characteristics that were specific to these HOX-expressing cells, and that is HOX expression is only in these MSCs. We don't see HOX expression in any other cell type, which of course we thought is a really big clue to what in fact HOX genes do, and that this HOX expression maintains its regional identity. So all throughout life you only see them in this regionally restricted area. They don't go to other areas of the body. And so Danielle asked one further question in this paper because as I told you before, we're using these HOX11 genes in the hope that this represents what all HOX genes do in other regions of the body. So in this very pretty particular experiment, she looked at the bone marrow stromal population from the radius and ulna, which I've just talked to you about, from the humerus of the arm, and also from the sternum and ribs. And if she looked at the bone marrow stromal component there, what she showed, and this is by qPCR, is that, as I've shown a lot of data on already, in the radius and ulna, HOX-A11 is expressed in that 5% of the LEPR positive stromal cells and not in the 95% of the LEPR negative. But HOX-11 is not expressed in either of those populations in the humerus, and it's not expressed in those cells in the sternum. But if you look at the humerus, what they do express only in the LEPR positive 5% and not the LEPR negative are HOX9 and HOX10 genes, which are the ones that pattern those regions of the bone. And similarly, if you look at the sternum and ribs, what you see is, again, in that LEPR positive fraction, you see HOX5 and HOX6 genes, which pattern the ribs, and not in the LEPR negative. So this really, I think, supported the idea that this is true of all HOX genes. HOX genes are maintained in these progenitor-enriched MSCs throughout postnatal and adult life in the mouse. So now I'm going to tell you about work that actually just came out in Nature Communications last week. So we just published this work that, based on Danielle's work, what we were wondering, remember our question started with these embryonically arising cells that of course aren't in the bone marrow because there's no bone marrow in an embryo. They're in this perichondrial region, and this expression is maintained through the whole life of the animal. So our question was, are these cells related to each other? Is this the same population throughout life? And could this be a skeletal stem cell pool that actually arises early in the embryo when the skeleton is formed and then is used throughout the life of the animal? And this was interesting to us, of course, simply as Hox biologists and being interested in skeletal biology, but it also suggested something that would be somewhat paradigm shifting in this MSC field, in that there are many publications that have looked at markers that also mark, to some degree at least, this MSC, including this progenitor-enriched MSC population. And what all of those genetic models have shown is that they can mark those cells in the embryo, but by adult stages, those cells are all gone. They aren't maintained through the life of the embryo. But if they start marking them at postnatal stages, now they can see them in a population that's maintained through adulthood. So this combination of results have suggested that adult MSCs arise postnatally and are different from the embryonic population. And this is inconsistent with what we were thinking of what we've seen for a Hox expression that starts early and appears to stay the same. So Kiriel, who defended in my lab last year, created this new allele that I'll explain a little further, this CRE-ERT2 allele, which allows us now not just to look at the real-time expression of Hox genes, but also to follow their lineage. And so this distinction between these two alleles is really important, so please stop me and ask a question if this isn't really clear. So in the HOXA11 GFP mouse I just told you about, we put this green fluorescent protein in place of the HOX11, and so what happens is wherever HOX11 is expressed, we now see a green fluorescent protein. When Hox expression goes away, the GFP also goes away. So that's to distinguish it from this lineage trace, which is, we instead now put a CRE-ER in place of the HOXA11 gene. This CRE really doesn't do anything by itself. You also have to cross in this lineage reporter. So this is the Rosa locus, which is expressed all the time in all cells. And it has a stop site inserted into it as well as now a red fluorescent protein. But this stop sequence in front of it, while this thing is expressed in all cells, or is there in all cells, isn't expressed because of the stop sequence. So what happens in combination with CRE is when CRE is expressed, and of course CRE is now only expressed in our HOX11 expressing cells, we can activate that CRE at any time, whether we do it in an embryo or a postnatal mouse or an adult mouse. We give tamoxifen to that animal at the time of our choosing. It activates the CRE, and when this CRE is activated, it deletes the stop sequence. And so now red comes on. We can turn red on in the embryo, we can turn red on in the postnatal animal, we can turn the red on at the adult stages, and now that red is only expressed where Hox is, but the difference between the red and the green is because this red is now being expressed at a locus that never goes off, it's not only expressed in the Hox expressing cells, it's expressed in all the cells that come from the Hox expressing cells. So it's a lineage label. It is expressed in the whole lineage of Hox expressing cells. So Hox expressing cells in anything that comes from it. So to validate this allele, first we could show it doesn't, I'm not showing you here, but it doesn't come on when we don't give tamoxifen. It doesn't come on in the wrong regions. This is the humerus of a mouse. It only starts coming on in the zeugopod, and it overlaps with the GFP. So it's being expressed in the right cells. And our experiment was, we were going to start this lineage label at both E13.5 and at P3, at postnatal day 3. So P3 is when other researchers have shown that you can start to label MSCs, and some of those MSCs you can find later in the adult throughout life. But that if you label at embryonic stages, this has never worked to label a continuous population through the adulthood of an animal. And what I also want to point out is lineage labeling at E13.5, where Again, obviously not labeling the bone marrow because there is no bone marrow. There's only these 2 cartilage rudiments. So if we see lineage labeling into the bone marrow, it proves or provides very strong evidence genetically that it's our Hox-expressing cells that have given rise to the bone marrow MSCs. So Curiel did this experiment, dosing at both time points, and hopefully what you can see here is that you see a lot of red in all of these pictures. That's the important point. These are pictures from animals that have been lineage labeled initiating at E13.5 in embryonic stages, and animals that have been initiated at P3 or postnatal stages. And we are looking 24 hours later, in this case 48 hours later, newborn stages, 2 weeks. This is an 8-week-old skeleton all the way out to 1 year of life. And what you can see is that whether we start the lineage label at E13.5 with 1 single dose then, or 1 single dose at P3, this continues to lineage label strongly in the skeleton throughout the life of the animal. And that these look indistinguishable from each other. So an E13 induction looks the same as a P3. Importantly, and this is particularly important with the idea of how MSC therapies can work, is that if you look at this animal any age, from if you induce it at E13.5 and take it at 8 weeks of age, a year of age, through the life of this animal, you will only see lineage labeling in the zeugopod. This is the same animal, and it's the humerus from that animal, and you can see these MSCs don't move. And so the idea of injecting MSCs and having them travel all over and find something, this isn't what MSCs do. They arise locally, and they stay locally throughout the life of an animal. If you take them and inject them into an animal, they stay right there. They don't go anywhere. They don't go rehome to other places. In this experiment, again, this is from animals injected at E13.5. This is from P3. Hopefully you can see they're virtually identical to each other. This experiment looked at a series of co-labeling experiments to show that our Hox-expressing cells do lineage label into cartilage, into bone, and into fat. So here's the cartilage layers you can see in an adult animal are just at the epiphyseal end of the bone anymore. You can see in the trabecular regions, in the beginning of the bone marrow of your bones, or along the cortical surface of your bones. These red cells are co-labeling with Asterix, which is a label for osteoblasts, the bone-forming cells. SOST marks these osteocytes, the bone-forming cells inside the cortical bone, and they also mark adipocytes, or the fat that develops in your bone marrow. So our Hox-expressing cells lineage label throughout life into all 3 lineages, showing that they're progenitors for all of these skeletal cell types. Importantly, also, this is if we induced at E13, wait till the animal's an adult, cause of fracture, we can see that this expanded lineage population becomes cartilage and becomes bone. So it is really serving as the progenitor for repair in these animals. And really critically, this is a set of FACS experiments that are, in this case, this animal was injected at P3. We waited till the animal was a year old and then said, are there still red cells in the bone marrow, and are those still MSCs? Are they still in the progenitor-rich MSC fraction, and are they still expressing GFP? So the answer is yes, those cells are staying GFP, and they're also staying fractionated into this progenitor MSC population. So these cells are self-renewing and being maintained as stem cells throughout the entire life of the animal. And then critically, this was also true when we gave 1 lineage label at E13.5. So this experiment really showed that these tiny number of cells that surround the cartilage element are in fact the MSCs that you have in your bone marrow when you are our age, some of us. So they are really stem cells throughout adult life. So when we attempted to publish this paper, one of the reviewers, I have to imagine just out of curiosity, said, well, that's really interesting, but we all know as animals or humans age, our MSC population goes down. So you really start to see less cells. We see less lineage labeled cells in an old animal than in a young animal. And any of us over 50 here are well aware that we have less stem cells now than we did when we were young. So what happens if you take an older mouse and then fracture it? So in this experiment, we initiated a lineage label at E13.5. We waited till the mouse was 10 months old, and then we fractured the animal and said, okay, are this tiny population of cells that we marked at E13.5, if we fracture the animal when it's pretty old, does it still lineage label to repair the skeleton? And hopefully you can see the answer was a resounding yes. That small number of cells really expanded to really serve as the progenitors for the entire repair of that skeleton, even when the animal is 10 months old. And again, contributed to both cartilage and to bone, both important for repairing the skeleton. So to conclude this part of the talk, what we just published and hope to convince the reviewers and any other readers of is that the Hox-positive cells are the skeletal progenitors. There's this self-renewing population of stem cells that contribute to all of the skeletal lineages throughout the entire life of the animal. And importantly, they self-renew throughout life. So they really are a stem cell population. They are the MSCs, or the skeletal stem cells, in the animal. And so this was very exciting to us. And now I'm going to just spend the last 10 minutes or so telling you about work that's going on in the lab now that is addressing the question of, it's great that Hox genes are stem cells. I think that gives us a clue of what they're doing. And I'm showing you that Hox stays expressed and stays in the stem cells throughout life. But again, to this idea that it's just an embryonic patterning factor. Does it stay functioning? Do Hox genes stay functioning in the postnatal adult animal, or are they just now marking it? And I showed you previously the dramatic patterning defects it has in embryogenesis, but does it continue to the Hox genes themselves do anything in these cells? So we had ruled out some possibilities of how Hox genes function in embryonic stages through the earlier work that Danielle did. And this, again, was somewhat surprising. So when we first found that these cells were in MSCs and potentially the stem cell population in adults, we thought, well, of course, maybe they might be in the stem cell population in embryos. And it would be pretty easy to imagine that if Hox genes controlled the maintenance of these stem cells, that when you lost the function of the Hox genes, you lost the stem cells. They couldn't self-renew anymore. You run out of progenitors and you can't make a bone anymore. So you end up with this little stumpy thing instead of your real bone. That was really probably the simplest thing that we could have predicted could have happened in terms of how Hox genes function. And that was not supported at all. So in this data, what we showed is that if you take the zeugopod region of this wild-type animal, or if you take this nubby little stump of this not very well-developed zeugopod in this mutant animal, and counted the number of GFP cells in it, it was the same. So the GFP cells weren't gone. So Hox was not controlling the ability of these cells to at least stay in the animal. And in fact, if you took the cells out of either one of these bones and put them into the CFUF assay in vitro to see do they still have progenitor activity in vitro, the answer is yes. That at least in vitro, they could continue to propagate and self-renew in vitro even without Hox genes. So that didn't support the idea that Hox was maintaining the stem cell population. However, when Jane Sung in the lab did a trilineage differentiation assay in vitro and asked, can these cells differentiate into bone, cartilage, and fat, the answer was no. They could differentiate into fat just fine. In fact, they made a ton of it for reasons we can't really explain. But they were really almost completely unable to make cartilage and really pretty bad at making bone too, which suggested maybe somehow Hox genes in the progenitor cells controlled the ability of these cells to make osteoblasts and to make chondrocytes that make cartilage. So to see if this was true also at postnatal and adult stages, Jane Song, who's a current graduate student in the lab, made this conditional allele in one of these two HOX11 genes, in this case HOXD11. In this case, she used these LOXP sites to flank the exon, one of the coding regions of this HOX11 gene. And in this case, instead of taking out a stop codon, we could take out part of the gene at any time that we wanted. By using the ROSA locus again, now the ROSA locus is expressing the CRE. So CRE is expressed in all cells at all times, but it's not active. But at the time of our choosing, we could give tamoxifen, and that tamoxifen would activate the Cre and then delete the HOXD11 gene. And it deletes the HOXD11 gene everywhere, but obviously that's only going to matter where HOXD11 is actually expressed. So using this conditional deletion, the really important thing when you create this mutation first is to make sure that you don't do any harm that you don't mean to do and that it works. So we put these LOXP sites outside the coding region for this gene, and the idea is that it shouldn't do anything. But that's important to check. And this shows here that with this conditional allele not recombined, these skeletons look the same as a wild-type animal. And importantly, we're going to cross this over, in this case, the HOXA11 null mice. When we put it over the HOXA11 null mice, which don't have a phenotype by themselves, you also don't get a phenotype with an unrecombined HOXD11 allele. So it's not causing harm. To prove that we are actually able to conditionally delete, and it works when we do that, we then gave tamoxifen to a very early embryo, right when HOX is coming on, to say if we're deleting that completely, we should reproduce the phenotype we saw in the null animals. And this also worked. So compared to the wild-type animal, when you conditionally delete at very early stages, you see this zygapod defect. So this really showed that our allele works. And we're able to ask this question now at later stages to ask the real question, which is, do HOX genes continue to function later in life? And so these are results that are currently underway in the lab, and we have some initial data that we think is interesting in suggesting, yes, that's the case. Our first piece of data, of course, we were excited to do the first experiment and started dosing right at the beginning of an adult stage of a mouse, gave tamoxifen chow for 3 weeks, and took those animals just 3 weeks later. And what we found in just that short of time is that compared to the bone marrow of the control, hopefully you can see this conditional mutant animal, this bone marrow is filling up with fat. That's a really abnormal response. If you look at the bone just next door, the humerus of the mutants and the controls, you don't see that phenotype in that area. So again, we're getting this region-specific conversion to fat cells. But what happens to the skeleton? And what doesn't happen to the skeleton, at least over the course of the first few months after deletion, is we thought maybe these mice would become osteopenic. That means that their bones would get smaller. They'd stop being able to make osteoblasts and cartilage, and you'd start to see a reduced size in the bones. That isn't what we saw. The bones really look about the same size. But when we did this Masser-Goldner stain, hopefully you can see a major difference between the controls and the mutants, and that is the bones in these mice, the cortical bones, are bluish, greenish. These bones are very pink. So what does that mean? And actually, when we first saw it, we were like, I don't know what that means. We asked all our bone biologist friends, and they're like, wow, I've never seen that in an adult animal. But what perhaps you can see in the control animal or in the wild-type animal is a little bit of pink staining at the edges of the bone and at the surfaces of the bone where the bone's undergoing remodeling. So this fusion stain stains osteoid material. So that's newly laid down bone that hasn't been put into a lamellar form, hasn't been processed so that the bone is long and tight and straight. And so what this suggested is that this bony material is not normal anymore. And just to show you the humerus from those same animals that were cut off and put into the same tube and stained the same way, they don't have this pink staining. So this, again, was a specific regional defect. So what has happened here is what we're trying to figure out. And here are some of the results, just the histology of these bones. This is just one section of the length of the lamellar bone along the length of, actually, of your ulna. What you can hopefully see are these striations of the bone that are long and straight, with the osteocytes, which are these internal bone-forming cells that are arranged linearly in conjunction with the collagen. What hopefully you can see in this mutant is that this histology is really sharply affected, and these osteocytes are no longer organized, showing that this bone material has really become different in some way. This is, again, the histology showing that this portion of the bone has become very mispatterned, and that using, oh my gosh, I just forgot the name of the stain. This red stain that I can't remember the name of, under polarized light and under bright field, This red stain stains collagen, organized collagen. You can see the osteocytes and their long projections and the striations that go a long way in a normal bone, and how you just see accumulated, aggregated collagen in these mutants. And by polarized lights, you can see this chunk of disorganized collagen that's replacing this lamellar bone structure. When Jane looked for differentiation of osteoblasts, what she saw is that the early markers for osteoblasts, this is osteopontin shown here in green, really doesn't look very different, although again you can see the disorganization of the bone. But osteocalcin, which is a really late marker for osteoblasts, is gone in most regions and maybe really, really faint in other regions, suggesting that while osteoblasts start to form, they can't mature in these adult bones. When we fracture these conditional mutants, what you can see is a callus does form in the mutant, but it's quite a bit smaller than the control. The callus is the expanding region of progenitor cells that are going to give rise to the cells that repair. And if you look closely inside this callus, what you can see is that, hopefully you can see here, is that Asterix is expressed in the control animals. This is, again, a marker for the initiation of osteoblasts. But there's no osteoblasts being formed in this fracture. And there's a quite reduced amount of SOX9, which is the marker for the cartilage cells, both cell types you need to repair the skeleton. And these cells look very abnormal, too, in ways we don't understand. If we look 6 weeks out after the fracture, you can see this is the two bones of the control animal, and this is the bone marrow. So instead of this gnarly material, it's all been remodeled, and you can see the bone marrow has been reestablished in this previously fractured animal. If you took an x-ray, you could barely see the line of this fracture anymore. In the mutant animals, you can see that this fracture is not repairing. It's not able to completely repair in this animal. So that is the stage we're at in this research now. And what they indicate to us so far is that Hox genes are controlling not the formation of the stem cells, they do mark the stem cells, but the stem cells' ability to differentiate into the proper cell types that are important for maintaining and repairing the skeleton. So both our embryonic results and our adult results so far are consistent with that possibility. So our conclusions to date is that Hox-expressing cells really are the skeletal stem cells. They're the cells in your body that are set aside at embryonic stages and are used as the stem cell pool to grow, maintain, and repair your skeleton throughout life. And this muscles that I showed in question marks, I took out of this talk, but in new work in the lab, these Hox genes are expressed in the skeleton, as I showed you, but they also have a continuous expression up in the tendons that attach to the parts of the bone they're expressed in, and in the interstitial stromal cells that are embedded throughout your muscle. They're not expressed in the actual muscle fibers, but in the muscle interstitial tissue. And when we lose the function of Hox genes in those interstitial cells, there are dramatic defects on the ability to maintain and repair your muscles as well. And Hox genes continue to function throughout the life of the animal. So whatever these Hox genes are doing, which remains a central question in the lab, we believe they're performing the same or similar functions from the time you're an embryo, through postnatal life, through growth, and in adult life for maintenance and repair. So this is continuing questions that we're asking in the lab. So to conclude, I'd like to thank the members of the lab. We're spread out a little bit all over the place. This is my new group in Madison, some of whom came with me from Michigan. Kiriel, who she went to Portugal to continue a project that was on a really short timeline related to this project for a year. And Jane is still back in Michigan finishing her PhD this year. Anna Miller is shown here. She just started in the lab in the last month and was not at this get-together. And I also want to give a shout out to the Department of Cell and Regenerative Biology that I joined. So these individuals are all assistant professors in the lab. They were all hired before that I came, and I have to say were a huge part of the reason I was so excited to join Madison. These people all study different aspects of regenerative biology, from pancreas to heart regeneration, neural regeneration, embryonic stem cells, and Ricky studies aging, another important process related to regeneration, as many of us know firsthand. And so it's with a really exciting group of young investigators that we hope to grow and maintain and continue this department. And so with that, thank you for your time, and I'm really happy to take any questions.
It looked on multiple slides, both the GFD and the lineage stating like there was preferential or greater expression in the distal than the proximal.
Definitely.
Is that an artifact or what's going on there?
No, it is distally biased. If you lineage label through time, it seems to go proximal. We don't really know why because the patterning is so restricted to the zygopod. But the strongest part, if you look at the Hox-10s, it's the same way, seems to be in this distal area, at least in the limb. It's been shown by others that most of your limb growth, this aspect of your it does happen distally. So your growth is preferential there, but not exclusive. So whether it is related to that, we don't really know. But that more distal expression is definitely what we really see.
If you look at the lineage stuff over time, though, one would expect that if it's functioning as a stem cell repair mechanism, it would be equalized throughout proximal and distal, wouldn't it not?
It becomes more proximal through that segment. So you can see the lineage label, is largely through the whole skeleton, but there is still really denser expression distally. It does become more equalized, but not completely. But it never goes beyond into the next element.
This is a comment. I found your verbal exposition very clear, but as a novice, as somebody who doesn't know this stuff, it's very difficult looking at diagrams or even pictures where there's more than, say, two
things
to compare.
Oh, that's helpful.
But your expression, very clear, very precise. Sometimes I just shut my eyes.
And listen, can't see all that at one time. Thank you.
And it worked just fine.
Okay. That's great. Well, probably not for the reviewers, but thank you. Yeah.
Yeah, I was just wondering, I know you went through this, I'm not familiar with it, but is there any practical things from what you know that can be applied, say, to help people?
Yeah, there's one really important thing that we published in this paper, in Danielle's paper actually, that's I think relevant. MSCs, as you may know, are being used for lots of different bioengineering and therapy purposes. And if you are a researcher studying how to make MSCs into any kind of material, and they'll make different organs or tissues, if you order MSCs from Lonza, is one of the big companies that sells them, they don't even tell you where the MSCs come from. In fact, most of the time they come from a hip. That's just usually the easiest place to get them, but they don't tell you where they come from. So I think one of the biggest implications of our research in terms of bioengineering and clinical use of MSCs is that we, I think, have clearly demonstrated that different Hox genes are expressed in different bone marrow cells. And so it really may matter, and now we're showing that they have a function. And certainly in the embryo, they have different functions depending on where they are. So this really suggests that where these MSCs come from may have profound implications on what you can do with them from a tissue engineering perspective, and might explain some of the reasons there's such variability in results that come out of these studies. So I think it's something that people should be paying attention to in terms of what is the actual pool of cells they're starting with. That's probably the most important implication translationally for this work.
If you were to take a group of Hox cells in an adult and transplant them to a different zone, do they survive? No.
Well, you can put them in, they'll stay in kind of a ball for a pretty long time, but they won't go repopulate. The Curiel in Lisbon is doing a hugely sophisticated set of genetic experiments to ask the question, what if you let the embryo develop properly and then switch the ones that are expressed? Does it matter? So that would really help answer the question do the different Hox genes have different functions throughout life like they do in embryogenesis, which would really reinforce the idea that it matters what population you start, or may well matter what population you start with. Mm-hmm.
Now, this question might be off the thing, but I know they're trying to come up and make cartilage.
Yeah.
And does this have anything to do with that because of arthritis and stuff like that?
These are the cells that would make that cartilage for sure,
Okay,
and we
so-
showed that as well.
So this is on the way of making cartilage instead
Yes.
of having a knee or hip replacement.
Right, that's right. These are the cells. It's the Hox-expressing cells, however few we may have left as we get older. That definitely are the cells that are going to be able to be pushed in the direction of trying, well, they are the cells that repair things, but to the extent we're not able to well anymore, it's that cell population that would be the progenitors in vivo that can give rise to
So
that.
you could inject some of these cells into somebody's arthritic hip or knee, and
Well,
it might...
This is the
I
problem.
don't know, I'm just
Yeah,
speculating.
the problem is that when you inject them, they can't go rehome. So unlike hematopoietic stem cells, which you can inject into an animal and have them repopulate, MSCs won't do that. They won't go set down again. So I think what you have to do is use the existing population and figure out how to stimulate them to regenerate in times and places where they're not anymore. Yeah, so it's more complicated, and it'd be great if we could just put them back in, but it doesn't, unfortunately, work. Yeah.
Would you put it back in, like into the knee?
You can, but they won't go re-adhere and do what they're supposed to do. They'll just kind of sit there in a blob. And I mean, they do this in a lot of surgeries, and it seems to help. But these stem cells are releasing a lot of factors that appear to be good for you, but they're not reincorporating into the tissue.
Could they be talking to the endogenous?
Could be. I think it could be. They don't really know. It does seem to help in many cases, but the current thinking is probably more they're just highly regenerative cells that are expressing a lot of goodies, and that probably helps. Not anything probably more specific than that, but it's not clear. Yeah. Mm-hmm.
And where do you look in the future as far as doing research on this?
For me doing research or in this field?
Yeah,
My passion is to understand what these Hox genes do. So I think that they're a fascinating set of genes, critically important for development throughout life. And I would like to be able to tell you 10 years from now when I talk exactly how they're doing that. I think we're learning some exciting things about what they do, but really are still at the beginning of trying to tell you how. That is what I'd like to do. Mm-hmm.
So as you get older, why do, let's say, your hip cells not regenerate as well as when you're younger?
So the main reason probably is that you lose a lot of your stem cell pool. So the stem cells maintain throughout life, but it really goes down. So those cells are decreased is probably one main reason, and the environment that they're in, probably because some of the stem cells, instead of self-renewing, just differentiate and then don't self-renew themselves. They also then start to create an environment that's a little more fibrotic, and that inhibits the cells that are still there from being able to behave normally. So it's sort of twofold, but those are the main reasons. Mm-hmm.
5 or 6 questions ago, you mentioned the idea that it's important to know where the cells are harvested from. Is that a fair?
Yes.
Can you tell me what ideally the information you'd like to show the provenance, if I may use that word, the source? What kind of information would you ideally get on a vial of cells so that you know what you got? How much of that is, well,
I think so. So in addition to being expressed in the skeleton, I should say that Hox genes are expressed in the stromal population of almost all your organs, not your brain, but the rest of them. So what I'd suggest for tissue engineering purposes is that you probably need to isolate stromal cells from the appropriate region of the body. So that it's those stromal cells that are most likely to be able to accurately be able to form what you're trying to form. There's lots of other things that need to be worked out, but probably starting with the right cell population is key. Is that what you were asking?
yeah, I wonder, you'd have to say it's from location?
Yes.
And then days of development, or?
Well, it shouldn't matter theoretically from what we know, of course. I mean, it really shouldn't matter what stage you take them from. Our indication so far really supports the idea that the Hox expressing population is really the same all the time. It's the furthest upstream progenitors that you can find. And so that doesn't prove, but provides some evidence that the important thing would be to start with the right cell population. It really might not matter so much when you get it. Embryonic cells tend to have much more regenerative capacity. They're just sort of more active. But that, from what we understand now, the cell population should probably be able to do the same thing as long as they're still there and they're still Hox expressing, which is something people don't look for in tissue engineering applications. They use this whole pile of stromal cells that grow out. So the other thing that might be useful translationally is to carve out those cells that are still expressing the appropriate Hox gene, because those clearly in vivo are the cells that really have and maintain the progenitor activity.
Now you're working mostly with animals. Has there been any work done in humans?
There has been. They've shown just recently, not in such a selected population of the stromal cells, but that both in the dermal fibroblasts and in the bone marrow stromal population, that they also maintain a Hox code that looks to be, as we would expect, the same as in the mouse. Because this work just came out in the last couple of years. It's really work people are looking at now. But there's no way to prospectively look at Hox expression in a human. It's a transcription factor that you can't, we can't genetically modify ourselves. And so there's no way to easily tell that prospectively when you're working with a human MSC population. Mm-hmm.
I haven't heard you mention induced pluripotent stem cells. Maybe that's because I'm completely off on that. But is there, what route, if any, might these have in addressing some of these origin?
Did you say IPS cells? Is that what you mean?
That's the right word,
Yeah, it is. IPS cells are embryonic-like cells, so they're cells that have been made to go back further than MSCs into an embryonic source of stem cells that can not only become MSCs, but lots of other different populations. So this Hox-expressing population only arises in mid-embryogenesis, and it arises sort of during organogenesis when you start to form organs. So the MSCs are downstream of the IPSs. It's interesting, we have a project that's just getting started with a person who does IPS. So he takes stromal cells, makes them IPS cells from humans, and then tries to redifferentiate them into musculoskeletal tissue. And what he's shown is that as they differentiate into MSCs, and this is human-like MSCs, the Hox expression goes through the roof. But not surprisingly, he has lots of different Hox expression because I don't think we know how, but certainly he doesn't know how yet, how you make them one set or the other, which happens in an animal, but not in vitro, because it just sort of makes all of them. Presumably, certain cells are making certain ones, although that's not even clear. But if you're going to use these even further back stem cells, IPS cells or ES cells, to do this differentiation, I think it's something you have to keep in mind is where are you differentiating that progenitor pool to first, which isn't something that's looked at, which is something that could be.
They don't have little signs on them saying.
Yeah, well you can, in that case, you can take a portion of those cells and then look. But if it's making all different kinds, it would be difficult to know how to sort that out, and if it could matter. But certainly Hox expression goes way up in those protocols as you get into the MSC stage.
So what's understood about during embryogenesis about what turns on the different Hox cells?
Yeah, what is understood? So retinoic acid is an important part of it, but it's a fascinating, I don't know if anyone's talked about epigenetics, but Hox genes are one of the key regulators of epigenetics. So in ES cells or IPS cells or in the very early embryo, all Hox genes are off. There's zero expression. And as gastrulation starts, which is when sort of the ball starts turning into a long tube and you start growing posteriorly, they come on one at a time. So first the HOX1 genes come on, and then the HOX2 genes come on, then 3, then 4, then 5. And as the embryo extends, all of them roll out in expression and then close down the other way. So that is going back to the first part of the talk of how you set up an AP axis and make this part of your body different than this part, then different than this part. This is sort of the key way you do this.
As the embryo is elongated.
as it grows out. That's when they come on. And so that's also as cells are starting to differentiate into, you know, the three major germ layers, the endoderm, mesoderm, and ectoderm. And so it's during that rollout when Hox genes come on and then come on in this very specific linear way, collinear way. Uh-huh.
In your slides, you compare your radius and ulna area to your humerus. How do you know when working with the embryo that you're getting it in just the right area?
Practice. Yeah. Practice, and you can also adjust those cryostats as you kind of start to go in. You move it till you're getting that just right section where you catch all of that in one plane is really just practice. Non-trivial. But important for how it looks.
Any other questions? One more.
If you took the Hox genes from an infant, from a specific area, like the knee, to use that as a progenitor for new bone?
Yes. Yeah, presumably. I mean, the trick is, I mean, yes, they definitely would be progenitors. They'd probably be great progenitors. The younger you are, the better you're at doing that. What is tough is to get it to integrate back into another organism. That's the part that it won't do by itself very easily. Muscles will pretty well, actually, but bone won't. So in terms of really regenerating, say, lost bone, you know, the problem is getting it to attach to what was there. Yeah. But definitely they would serve as a good progenitor source from everything we know, for sure.
Thank you very much.
Thank you.